Okay, can somebody confirm you can hear me?
Can hear you.
Great. Get out of here. My computer's going to be a bit slow because it's still processing the lecture page. So just please bear with me. Oh, come on. All right, you should be seeing my web screen now. Nothing really here. I did not get much of a labs graded from last week, so I'll try and work on that tonight. And if I don't succeed in getting the lab graded tonight, I will make it so the second turning in will not be due this Saturday, but make it due the following Saturday. Let's see. All right, today is 8/6, and we should be covering Lab 7, throat and urine cultures. Tonight, sometime tonight after the lab, I'll be sending you the first data and your unknown number for the unknown project. And if you don't understand this project, you'll have to take a look at the discussion that was on July 30th. And I was a bit late putting that up. Sorry about that. It wasn't until somebody asked for a lecture on July 30th that I realized I hadn't posted the lecture and lab on July 30th. All right, let's go to lab 7. Any questions? I think I can close that out. Oh, let me state that some of you aren't doing too well in the lab, and it would help you if you would stay in the lab. And as you're working on the questions, if you have a question, to ask me. Lately, students have been leaving right away, which isn't a problem for me. That means I can log out sooner. Let's go to module 7, throat and urine cultures. There is some reading, but not too much, a page on nose, throat, and mouth normal microbiota. A little bit about beta hemolytic streptococci, actually in the end of this lab in the appendix. The same with Haemophilus in the appendix. There's one page to read about the oxidase test, another page to read about the catalase test, and then coagulase production. There's a little bit in the appendix of this lab module talking about coagulase. In this lab, there's some supplemental materials. You'll watch some video clips. The learning objectives in this lab will be: one, be able to explain how to properly collect throat and urine specimens for routine laboratory culture. And then two, list some common bacterial pathogens isolated from the throat and urine cultures. Three, know, know the culture media used to cultivate these pathogens. Four, recognize which tests are used to identify throat and urine pathogens. And then five, and lastly, understand the principles and how to interpret the following tests: the catalase test, the coagulase test, and the oxidase test. We will talk about the XV factor testing, but I don't expect you to understand it, and I won't ask any quiz questions about it. We'll just briefly talk about it. Oops, too far. So once a specimen is received in a lab for growing them, they're logged and processed in a timely fashion. Specific culture media are inoculated and incubated depending on what the source is. There are throat and urine specimens we're going to talk about today. Below is an overview of the culture media used to cultivate common bacterial pathogens isolated from throat and urine cultures. For the throat specimen, for adults, There's really only one culture media that needs to be cultured, and that is blood agar, where we're looking mainly for Streptococcus pyogenes, a common bacterial pathogen found in the throat. But for children, especially small children and infants, we also use blood agar and chocolate agar. And then we're looking for, yes, Streptococcus pyogenes, but we're also looking for Haemophilus influenzae in young children and infants. Any question about any of that? And those are the only common bacterial pathogens isolated from the throat. In urine specimens, we have different media we use, and there's different common bacterial pathogens. We always use blood agar for the culture media when we have a urine specimen, but we also use either McConkey's agar or EMB, eosine methylene blue agar. And we'll talk about why we use one or the other, but we always use one or the other and blood agar. And then we're looking for various Streptococcus species, Enterococcus species, Staphylococcus species, and then members of the family Enterobacteriaceae, especially E. coli, as well as members of the family Pseudomonaceae. These are all common bacterial pathogens isolated from urine specimens. Any question about any of that? All right. Sore throats may be caused by viruses or bacteria. If the clinician finds white patches on inflamed tonsils or the back of the throat, whitish spots, oops, good. The lecture Zoom file just finished, so my computer will be working a little better and faster. If we find white spots on the tonsils or the back of the throat, perhaps on the tongue, but that's not as clear, that is from a bacterial infection. If, on the other hand, the back of the throat and the tonsils, as well as the tongue, are only reddish, that's usually indicative of a viral infection. In this lab, we're only going to talk about bacterial infections. The most commonly isolated bacterial pathogen, isolated from the throat, is Streptococcus pyogenes. And this is the bacterial species name, what I call it. It's also called Group A Streptococcus, which I don't use, because although that is the same species, that's not a species name. And sometimes it's abbreviated as GAS for Group A Streptococcus. Strep throat is isolated by swabbing the back of the throat or the tonsils where there is redness and or white patches. You want to avoid touching the tongue with a swab, the sides of the mouth, the top of the mouth, or the teeth, as well as the cheek, I guess. Well, that's the side of the mouth. And you usually use a tongue depressor to help swab the back of the throat and the tonsils. If the specimen is collected properly, patients are then tested positive for Streptococcus pyogenes, are placed on antibiotic therapy. The reason being is that Streptococcus pyogenes has I'm gonna get it, capsule around it, which makes it very difficult for the patient to throw off the bacteria infection. Oops. And there's some test kits. We're not going to talk about those for Streptococcus pyogenes, but because Streptococcus pyogenes is fastidious, the culture medium of choice is blood agar. That is TSA supplemented with 5% sheep's blood. And then the red blood cells supply the organ, the chemicals or not organisms, but chemicals or nutrients that Streptococcus pyogenes requires to grow. And then you look at the blood agar plate and examine it for any small clear colonies surrounded by a complete zone of hemolysis. And the colonies are actually whitish. Any beta hemolytic colonies could be Streptococcus pyogenes or some other bacterium. You only need 2 biochemical tests required for definitive identification of a beta hemolytic bacteria growing on blood agar isolated from the throat. And that is, you need to do the Cadlase test and the bacitracin susceptibility test. So here we have bacteria, and that's actually the normal throat where there's no hemolytic activity there. Let me blow that up a little bit. It looks like there's some alpha hemolytic activity here. And then right there, we can see one colony with alpha hemolytic activity. There's probably some others, but one very obvious. Most of them are not hemolytic. And just call this over. And there we have our bacteria colony right there. And it's surrounded by a clear zone of agar that is complete hemolysis. This is beta hemolytic bacteria isolated from the throat, streaked out on the plate. And there you can see some beta hemolytic colonies there among colonies which are not beta hemolytic, which we can see one there. That's actually alpha hemolytic there. Let me shrink this down a little. There we go. So the 2 tests we need to do, Cadlase and bacitracin. What you do is with Cadlase test is you put hydrogen peroxide on a slide, and then you add the bacteria. If the bacteria have Cadlase, they will produce water and oxygen from the hydrogen peroxide, and you'll look for bubbling, which is the oxygen being produced. If the bacteria test positive for Cadlase production, you see bubbles of oxygen produced. Streptococcus and Enterococcus species test negative for Cadlase production. So they will be negative, and positive would be Cadlase positive, would not be Streptococcus pyogenes. You then do the bacitracin test, meaning this is something that was blood agar, beta hemolysis, Cadlase negative, and you streak it out on the plate here, putting down a disc of bacitracin on top of the growth, and there you can see the bacteria, and there is a zone of inhibition, which is a positive test for bacitracin, and on this one here, we have no zone of inhibition or very slight zone of inhibition, and that is negative for bacitracin, and that is not Streptococcus pyogenes. Streptococcus pyogenes is sensitive to bacitracin. Another organism that is clinically significant when isolated from the throat of children or infants is Haemophilus influenzae. It is a gram-negative coccobacillus. Haemophilus influenzae is one of the common causes of ear, eye, and or sinus infections in babies and young children. Haemophilus influenzae is fastidious, a tiny gram-negative coccobacillus that requires the growth factors, NAD, that's the V factor, and hemin, the X factor, which are supplied in chocolate agar. And so if you grow Haemophilus influenzae on chocolate agar, you'll get growth. If you grow it on tryptic soy agar or blood agar, Haemophilus influenzae will not grow, with one exception, and that is you could, I blow this up, grow it on blood agar if you have a streak of Staphylococcus aureus on the blood agar. So here we have our blood agar, and we did a single streak of Staphylococcus aureus, and here we did streaking of Haemophilus influenzae across and perpendicular to the blood agar. You will note, let me blow that up a little more, that the Haemophilus influenzae is growing near and around the Staphylococcus aureus, but it is not growing further away from the Staphylococcus aureus. And what's happening is the Staphylococcus aureus is lysing the red blood cells, which isn't shown here. Maybe it is. Let me pull it up way. Hopefully I'm not moving the picture. Come on. Yeah, I didn't move the picture. Oh, well. Actually, it is shown here, and that is there's the streak of Staphylococcus aureus, and then the clear zone is the hemolysis of the red blood cells, and that's around the streak. And what happens is the Staphylococcus aureus is lysing the red blood cells, and then the chemicals from the red blood cells are diffusing out from this zone hemolysis out to here. And then the hemolysis, the Haemophilus influenzae grows in this region here, but only near the region of Staphylococcus aureus. And it's because the Staphylococcus aureus is lysing the red blood cells and allowing the ingredients required by Haemophilus influenzae then diffuse out to here. But beyond that, the Haemophilus influenzae, like in this region here, is not growing. And that's because the stuff being released by Staphylococcus aureus from the red blood cells is not diffusing out here. Any question about any of that? So normally we just grow it on chocolate agar. Chocolate agar is the same as blood agar. The difference is you add the blood when the media comes fresh out of the autoclave. And then that's so hot that it lyses the blood, making the agar look brownish. And that's why it's called chocolate agar, because it looks like they added chocolate. They actually added blood at a very high temperature, which simply lysed the blood. When you lyse the blood, then the ingredients that the Haemophilus influenzae require are present, and Haemophilus influenzae can grow on chocolate agar. Any question about any of that? About there? All right. Urine cultures are diagnosed to test for urinary tract infections or UTI. That can be obviously urinary tract infection in the urethra, cystitis, the bladder, urethritis, that's the urethra, or polynephritis in the up as far as the kidneys. Normally, urine in the bladder is fairly sterile. Actually, in female bladder is no longer known to be sterile. The female bladder, at least the female bladder, has a normal microbiota. And I don't know, no one's ever looked at males to see if males have a normal microbiota in their bladder. Bacteria, however, can travel through the urethra up to the bladder, causing a infection of the bladder. Females are more predisposed to developing a UTI than males simply because females have a shorter urethra than males. To sample, the specimen for a urine culture, you first clean the genital area, and then patients prefer a collection that's called clean catch midstream, where you first begin voiding the urine and you don't collect at the beginning of the stream or the end of the stream, you collect the urine midstream. This reduces the transient organisms that are obtained from the urethra, meaning the urethra does have microbes on it. And if you do the clean catch midstream, you'll just reduce the presence of the microbes from the urethra. They'll still be there, but they'll be reduced. A better way is to do catheterization, where you stick a catheter into the bladder and then sample the urine directly in the bladder. The trouble is the catheter has to go through the urethra, and it can pick up microbes from the urethra and then put it into the bladder. And so you could still get microbes even if you do catheterization. And then I guess the best way would be to do a suprapubic aspiration, where you stick a needle through the abdominal wall directly into the bladder and then sample the urine directly. Obviously, you sterilize or clean the skin before you put in the needle. Patients, of course, do not like the suprapubic aspiration. They would much rather do the clean catch midstream. Those are the three methods you can collect urine. You then take the urine and put 0.001 millimeters, that's 1 microliter of urine, onto a blood agar plate. And we actually have a loop shown here that right there, you dip it in the urine, it only holds 1 microliter or 0.001 milliliters of urine, and then you put it in the on the blood agar plate. We use blood agar because it can grow both gram-negative and gram-positive organisms, as well as it can grow many fastidious organisms. And like which Streptococcus pyogenes is not normally found in the urine, but it could grow on blood agar, is my point. If it's just the fastidious organism I know that will grow on blood agar and will not grow on general purpose media. And I can't think of a fastidious organism that comes from the urine normally, but Streptococcus pyogenes is one which could grow in the urine, but is not likely. And besides growing on blood agar, we also take the urine and streak it on either MacConkey's agar, that's also called MAC agar, or EMB, eosine methylene blue. The reason for growing it on one of these two agars is that commonly the family Enterobacteriaceae causes, actually, I think it causes the majority of UTIs. And then the single most cause of any species causing a UTI is actually E. coli. And family Enterobacteriaceae, as well as E. coli, will grow on MacConkey's agar or EMB, and gram-positives will not grow on MacConkey's or EMB. So that's why we grow both on blood agar and either MacConkey's or EMB. We grow it on MacConkey's or EMB because most UTIs will be from the family Enterobacteriaceae, which will grow on the MacConkey's or EMB. But there are some gram-positives which will not grow on MacConkey's, which cause UTIs, and so that's why we grow it on blood agar or BAP plates. Interpretation for a positive urine culture is multifaceted. Factors that need to be considered are the type of collection. Is it voided versus catheterized specimen, meaning the catheter will have less contamination of bacteria from the urethra than the clean catch midstream. The gender of the patient, I'm not sure why, but actually females have more contamination of bacteria from the urethra than males. And then the number of colonies per mil per urine, the presence of microbial growth. If there's multiple organisms isolated from the UTI, then that could be indicative of a poor collection technique. Males have around 500 colonies per mil of urine. Females are greater with around 100,000 colonies per mil. And, this might be not necessarily the contamination from the urethra. It could be that females have probably more microbes growing in their bladder than males. Males might not even have a normal microbiota in the bladder, and nobody's ever looked, so I can't tell you, but that might be why there's a difference. All right, some tests that are done on the bacteria that then grow out of the urine culture is the coagulase test. Coagulase is looking for the enzyme coagulase, which is used to differentiate Staphylococcus aureus from other members of the staphylococcal family. The other members of the staphylococcal family are coagulase negative, and Staphylococcus aureus is coagulase positive. Coagulase actually contributes to the pathogenicity of Staphylococcus aureus, converting blood plasma fibrinogen into the protein fibrin, which allows the blood to clot, and that protects the bacteria from phagocytosis, being eaten by white blood cells, when the bacteria are few in numbers. So Staphylococcus aureus can create a blood clot, which then makes it harder for the immune system to attack the Staphylococcus aureus when the infection is initially getting started, when the bacteria are few in numbers. There's two tests for the coagulase test. You can test the coagulase on a slide where you put the bacteria cells in the coagulase, and if the bacteria have the enzyme coagulase, they'll create clotting or clumping, and if it's coagulase negative, it will be smear, uniform liquid smear. There's the test tube test, where if the bacteria create coagulase, the serum will clump or clot, will not pour. And then the coagulase negative test tube test, you tilt the tube and the serum will be liquid. The most common bacteria isolated from routine urine culture is E. coli, and these belong to the family Enterobacteriaceae. One test to use to separate the family Enterobacteriaceae from the Pseudomonaceae is the oxidase test. Pseudomonas aeruginosa, as well as other members of the Pseudomonas family, are gram-negative bacilli that may cause a UTI, as well as the family of Enterobacteriaceae can cause a UTI. They're both gram-negative bacilli, but the Pseudomonaceae are all oxidase positive, and the family Enterobacteriaceae are all oxidase negative. The Pseudomonaceae are also all aerobic and non-glucose fermenting species, and the family Enterobacteriaceae are all facultative anaerobes, and they can all ferment the sugar glucose. The oxidase test is looking for the presence of cytochrome C, which is an important protein in the electron transport chain. So if it isn't performing aerobic respiration, it has to have cytochrome C, and the Pseudomonaceae have cytochrome C, and that's why they are oxidase positive. All right, I already stated that. Oxidase test, you simply put the ingredients down on the cells, and if the cells are oxidase positive, within 20 or 30 seconds, the area will turn purplish or a dark blue, which is oxidase positive. If there were cells over here, this would be oxidase negative. Any question about any of that? All right, the urine and throat cultures are two of the more common types of specimens received in a lab for bacterial cultures. Microbiologists will grow them up and then test to see if the patient has a common bacterial pathogen. All right. So you'll need to view the following videos and answer the questions not in the lab module, but in the worksheet. You're not physically going to be performing any of these activities. However, you will be responsible for knowing how to perform them. Everything we do will be done online, so you won't actually be doing it. But there's a little video on running the catalase test. Watch that video. There's a little video on running the coagulase test. Watch that video. And there's a little video on watching the oxidase test. Let's go to the worksheet now. Whoops. So answer these questions. And the worksheet does have different questions in the module. Obviously, do not submit the module. If everybody were to do that, my web page would become full, and then you would no longer be able to turn in an assignment. If you turn in the lab module the first time, you'll get a warning. The second time, I'll give you a penalty. So question 1, answer which of the enzymes, catalase, coagulase, or oxidase, is the correct answer for questions 1 through 3. Which enzyme converts fibrinogen into fibrin? So that's 1A. 1B, name a microbe that tests positive for this enzyme. 2A, which enzyme detects the presence of cytochrome C in the electron transport chain? 2B, name 2 microbes that test positive for this enzyme. And then question 3A, which enzyme hydrolyzes hydrogen peroxide into non-toxic products, water and oxygen? And then 3B, name 2 microbes that test negative for this enzyme. Test 4, which rapid test used to differentiate between the following? I mean, these are the rapid tests, the catalase, coagulase, and oxidase tests. So 4A, which enzyme can differentiate between Staphylococcus and Streptococcus? 4B, which enzyme can differentiate between Staphylococcus aureus and other members of the Staphylococcus family? And then 4C, Enterobacteriaceae, such as E. coli, can be differentiated by this enzyme from the family Pseudomonaceae. So which enzyme is 4C? All right, 5. Concerning specimen collection for routine throat culture, what areas of the throat should be swabbed? 5B, what areas of the mouth should be avoided? 6. What is the common cause of bacterial throat infections? And list the species. Don't list the family. list the species. Question 7, what test is used by microbiologists to differentiate between beta hemolytic group A Streptococcus from other beta hemolytic Streptococcus species isolated in throat culture? Meaning we've done a throat culture, we grew it on a blood agar plate, and we find beta hemolysis. So which test do we need to run to separate out group A Streptococcus from other types of Streptococcus species? And then question 8, answer the following concerning a child's throat culture. 8A, state the 2 non-selective agar plates that are inoculated, one here and one there. And then 8B, what organism can be isolated on CAP but not on BAP? BAP being blood agar, CAP being chocolate agar. 8C, I've crossed out. You don't need to answer it because it's not discussed in the lab module. If you do want to answer it, you don't get extra credit, but you can look it up online. And then 9, list 2 media that are used to grow urine cultures. So we always take a urine culture and grow it on 2 different media. And then 10, list 3 reasons why urine specimens should be rejected for culture. And that's discussed in the lab module. And then 11, how much urine is placed on the surface of a blood agar plate from a urine culture? And then 12, what is the most common bacteria species isolated from patients with a bladder infection? So you need to name one species. And then 13, identify the Gram reaction, that's the Gram stain, and the cell morphology, the shape, not the cellular growth arrangement of these bacteria. 13A, Staphylococcus saprophyticus. 13B, E. coli. 13C, Pseudomonas aeruginosa. 13D, Streptococcus pyogenes. And then 13E, Haemophilus influenzae. All right, any questions? If not, go ahead and get started on the lab. I will be here until the last student logs out or until 9 o'clock, depending on which happens first. All right, go ahead and list your question.
Yeah, my understanding regarding to the blood agar, I thought it's an enriched general purpose media, which is non-selective. But I think you just mentioned it during the lecture that it is actually selective media.
No, blood agar is not selective.
So it's non-selective.
It is non-selective.
Okay, got it. Thank you.